Welcome back to our second session of the first day of the Human Cell Atlas Latin America Symposium 2022. This morning, we had 5 great talks covering different aspects of single-cell transcriptomics. Now, in our second section, we will start with a talk about the Human Cell Atlas data platform, as well as we'll have other 4 lightning talks from PhD students and postdocs developing amazing works on single-cell genomics and bioinformatics. Finally, we will finish our day with some breakout sessions to discuss important aspects related to single-cell research in our region. This afternoon will be moderated by myself, Vinicius Maracajá Coutinho from Chile, from the University of Chile, as well, and also by Dr. Yesid Cuesta from Colombia. Now, I would like to introduce and welcome our next speaker, Dr. Enrique Sapena Ventura from the European Bioinformatics Institute. Dr. Enrique studied biology at the University of Valencia in Spain, where he also did his bioinformatics master degree. He's now currently working as a data wrangler for the Human Cell Atlas Data Coordination Platform Ingestion Service, where he works maintaining the HCA metadata schema and acting as a data curator for submissions that are of interest to the HCA. This work has helped him to gain expertise that ranges from single-cell data and metadata curation to data management and verification of data. Enrique, welcome, and thank you very much for being here with us.
As Vinicius introduced me, I am Enrique, and I work for the Human Cell Atlas Data Coordination Platform, specifically for the ingestion service. It is a bit of a mouthful, I know, but I'm going to start sharing some slides. So what I want to speak to you about today is about this data coordination platform, what we are, what we do, and how we can support the HCA community. I'm going to start briefly by stating a little bit what is the HCA community. So the Human Cell Atlas community is just you, basically all you wonderful scientists doing all the research, doing all the scientific breakthroughs, and profiling the millions of diverse human cells that the Human Cell Atlas wants to have to create a comprehensive map. This is, of course, a global effort that requires hundreds of labs, generating data about specific organs and using a lot of a diverse range of experimental techniques and data types. So this community is very diverse, right? As the cells that we want to profile. So the first thing to look at is what is this community composed of? So this community is composed of the experimental scientists, the people that are on the wet lab, generating the data that want to find and visualize data of interest, the computational researchers that want to find more data to add to their own data sets, or just to do a review and to analyze and test hypotheses. And on the other end, we also have the engineers that want to download HCA data and create and test tools to ultimately contribute them to the Human Cell Atlas community. So one thing that we need to think of is, with this big community, with this diverse community, how do we bring all this data together so that everybody in the community can find and reuse it? The first thing to think of when we think of a platform that to do that is that platform needs to be comprehensive. It should have all the types of data that are needed to create this map. It should also be inclusive, and that's thinking about the talks that we've had in this morning. This is especially important for the HCA Latin America meeting, because we want data from all over the world. We want that diversity. We want that in. The data needs to be organized so that it is easily accessible. It is standardized so that it's understandable. It's also the platform also needs to be accessible for everybody. And of course, single cell being the evolving community that it is, it's a somewhat recent technology. We also need to be dynamic, and we need to adapt to the needs of the community. So we've talked about what this platform must accomplish, and I'm going to speak a little bit more on how we are making it possible. This is a small bit of the overall flow of data in the data coordination platform. So the first thing is the laboratories are generating that data, right? And they want to contribute with it. We, in the data coordination platform, help them curate and upload that data into the platform so that then that data becomes publicly accessible to other researchers. And also, we also help for the tool developers and for the people that are looking for tools to analyze their data. We also help by showing those tools in our portal. It all starts with the contribution step. So the first thing we do is with the help of a data wrangler such as me, for example, we maintain communication with the collaborators, and we assess the suitability of the data, of course, being the Human Cell Atlas. Right now, we are not accepting data from, let's say, pig. We do some legal requirements assessment as well. Currently, we only accept data for, with open access requirements. That is to change in the future, but for now, these are the requirements. And then, yes, we use this high standard, like metadata standard and control vocabulary to annotate the data so that it can be easily interpretable for the other researchers later. And we also help curate the matrices to be compatible with cell by gene and other data visualization portals. We then review, like upload the data, and in the end, the data becomes publicly accessible in our portal. And we also provide with accessions for the data and the metadata to be used in the publications. So the data is already in the portal. Now, what do I do to find it? So in the portal, and I'm going to do a small demo, live demo, hopefully it goes well. I'm going to do a small demo, but basically you can use a lot of the metadata, the metadata that we have curated in these first steps to search for that data. So you can find, for example, data from humans that in between the ages of 10 and 19, let's say. You want also some kidney data. All that is filterable so that you can find that data and create that cohort of your interest so that then you can download that data. And it's very easy. You'll see it in a second. And you can either download that data or export it into Terra, where you can automate your workflows on the cloud. I only have 12 minutes, so I'm not going to go into the whole Terra thing. But if you are interested, please let me know, and I will point you to the point you in the direction of people that do know more about the Terra data portal. So yeah, let's go into the data portal. As you can see, very easy. We'll just go into the explore page, and we're going to try to find some brain data from human, and that the library preparation technology that was used was either 10x 3' version 2 chemistry or Visium. So we will start. We can either do a free text search or just search in the filters that are here. but we're gonna do a free text search. So first of all, human, so Homo sapiens. We're going to do brain, and we're going to filter by the next prime, B2, and or BSIM. We'll see that we have 7 projects. If we only wanted, let's say, BSIM, we could just, because this is an OR, so if we only wanted BSIM, we can just clear this filter by clicking in the filter. And we'll see that we have one data set with brain data that contains spatial transcriptomics data. And then we can just go to either to the project by clicking on the name, or just to export selected data. And this will bring you to a page with a little bit of summary on the data. And yeah, and some instructions. You can either download the data requesting the curl command. This is going a bit slow, but basically here you will find a summary of the data that you can download. Oh, actually, no. Sorry. First, it was confirming the species. Yes, I always forget about that. And then you find the summary. Let's say that you just want the raw data, and that's it. And you can choose between Linux or a download option for Windows. And that's basically it. And if you want the metadata, it's just requesting the file manifest, or just in here in the main page, you can find the metadata just by clicking here, or the matrices just by clicking here. It's very easy, and I encourage everybody to use it. It's really easy. It's completely free. Like everybody can use it to download their data and or search for some data and tools. And yeah, as I said before, our data portal also links to the community portals and the tools, so anybody can also contribute. The slides will be shared later, if I'm correct. So there is a link here, so you can contribute if you have a tool of interest or any portal that you want to highlight. We're very welcome on that. And yeah, we work with many different portals like Sail by Gene, UCS CSL browser, the Single Cell Expression Atlas, the Cambridge Cell Atlas, and with some package and resources, with some package resources like Bioconductor, for example. And yeah, this is our portal, and our data is growing a lot on each monthly release. We are very happy about that. We are doing our best to capture all this diversity that we want to capture from, like, that we wanted to capture, but still there's a lot of diversity that we need to get. For example, I did a small quick research on the datasets that we have, and I noticed that we only have one dataset that contains a contributor from Latin America that is specifically working in Latin America. So we need to change that. We really need to change that. We need this community to bring their data, show that HCL Latin America is growing strong in the single cell field. And yeah, just a little bit on our future plans. So we are working, as I said before, in showing managed access datasets. At least the raw data, the count matrices we sometimes have depends on the legislations, basically. And we are also working with the Atlas builders. Such there are some consortia and like funding going around about generating atlases. So we are trying to work with them. One example would be the BioNetworks, and we are working with them to ensure that we are capturing enough metadata and that we're capturing the data that they will need to create these atlases. We are also collaborating across consortia to promote some metadata standards for the matrices. And this is part of our work as well with Cell by Gene, trying to get a format for matrices that is reusable, that is fair. And also we are working on streamlining the data contribution by allowing programmatic submissions. Currently, the submissions are only allowed via Wrangler and through a UI. But we are working to make sure that it is more accessible for everybody and that it's easier for people that would prefer to go through a programmatic submission route. So yeah, that's it. We welcome your feedback on everything from submitting the data and the data portal, discovering that data. What are we missing? Is there any feature that would be of interest to the community? And we welcome your feedback in any of these channels, either to this email for general feedback on the DCP community on Slack, and of course through if you have any data that you want to contribute, we are very happy to help, and you can just send an email to wranglerteam@data.humancellatlas.org. Lastly, just a little bit of an acknowledgement on all the institutions that we work with. Without them, this will not be possible. And of course, the team that are working in these institutions, they're all great. They're all doing a great job trying to make this possible. And yeah, shout out to all the partnership and consortia that are funding this initiative. Yeah, thank you.
And we have a question here from Gamal Al-Akabani about the common coordinate framework that before you answer, Enrique, I'd like you to, if you can explain a little bit to the audience, what is the common coordinate framework? And the question is, I would like to know if there has been any progress in developing algorithms for the common coordinate framework.
So I have to be completely sincere on this one. I have been in some conversations with this CCF, with the Common Coordination Framework, but they are not developing as part of the data coordination platform. They are their own. This was a very long time ago. I'm sorry, I cannot provide with a good answer here. I can, if you send me your email, or if you send an email to me, Gamal, I can point you to the people that will be able to answer this question. But unfortunately, I cannot provide much about the CCF.
Thank you. Thank you, Enrique. I have another question here from Yasid Cuesta. According to the different type of data you manage with this new type of data that you're managing in single cell transcriptomics, he's asking if it's there a new type of ontology, like gene ontology, specifically to manage single cell data.
So that is interesting. So the in the HCA, we use ontologies to define several pieces of metadata. And ontologies, just to put it very bluntly, is just a form of control vocabulary in which all the terms also have a hierarchy and it helps define, right? So for example, we have the gene ontology. The gene ontology is defining genes, but we also have Uberon. Uberon is defining body parts. Mondo is defining diseases. In this specific case, I think you are thinking about the cell line ontology, which is an ontology that is trying to generate terms, like unique terms, identifiable terms for all the cell types that are out there. And my answer to that is that we're working with them. We have, I think it's bi-weekly meetings with them where we discuss development on that and what should be the best practices, what terms should be added, how should we. One of the things, for example, is that there's a lot of discussion around how do you really classify like a cell type in a term that will not be reusable, because like using like somebody may, like some researcher may say, oh, this is a T cell, and some other may say, oh yes, but this is a T cell, blah blah blah. So there's a lot of discussion about that. But yeah, the cell line CL ontology, I think it's what you're looking for.
Quite complicated. It's a new word.
Thank you.
Yeah, thank you very much, Enrique. Now we need to move forward. If you have any questions to Enrique, you can let it on the chat, and Enrique can answer it by chatting. I would like now to invite Yacid Cuesta to moderate our next session, the lightning talks. Welcome, Yacid.
Thank you, Vinicius. So yes, as Vinicius said, it's time to start with our lightning talks. We are going to have 4 lightning talks from different topics. So the first lightning talk is by Candelario Hernandez.
The title of the talk is The Role of Copy Number Variants in Gene Co-Expression Patterns for Luminal B Breast Tumors. And the authors of this work are Enrique Hernandez Lemus, Jesus Espinal Enriquez, and me. And we work at the Instituto Nacional de Medicina Genómica in Mexico. This is the outline of my presentation. Very quick, I'm going to talk about this, that is the molecular classification of breast tumors. First, we have a luminal A and luminal B that are estrogen positive subtypes. They have positive receptors to estrogen, and then we have a HER2 that is certain tumors that have an amplification of this gene that is located in the 17th chromosome. And the triple negative means that this kind of tumors doesn't have estrogen positive receptors, and the HER2 gene is not altered. So just to understand a little what we are doing, I'm going to show this, and that is a way we start from matrices of expression and find and obtain a matrix of co-expression. We have here the expression matrix. You can see that this G1, G2, and so on are the genes. And these are the samples, the 1, the 2, the 3, and so on. We can have hundreds of these samples, and we can have all the genome in this site. And we can do a correlation matrix using the Pearson correlation in this way. I have the matrix of correlation here. And it's important to know that some values have no sense. For example, the values in the main diagonal are trivial, and these are repeated. So the matrix is symmetric. But if we arrange all the other numbers, we can find a certain of Gaussian curve, and this Gaussian curve is going to be kind of identifier for this matrix and for the network. So it's important to keep in mind, because in our work we do a lot of these networks and classify it using that distribution. So we have the adjacency matrix, and from the adjacency matrix we can build the network using this procedure, but instead of Pearson correlation, the team used this procedure to obtain with mutual information this important result. We can split this result or this figure in two parts, 2 times. The first time we are going to split it in a left and right. If you look the left side, you have a lot of connections that belongs to healthy tissues or healthy samples. In this case, in the A case, we have all the genome, and in the C case, we have the chromosome 1 and chromosome 19 links. And in the other side, we have the same, but for cancerous tissues. In this case, there is no classification. We have cancerous tissues without classification of all kinds, luminal A, luminal B, and so on. And if we split it in up and down, is what I say. We have the whole chromosome, the whole genome, and here we have chromosomes 19 and 1. And you can see that the connections are really strong and long distance in the case of healthy tissues, and all that long links or correlations are lost in the case of cancerous tissues. So the question is, what is the reason because of this? Many people suggested that this loss of correlation was due to the copy number variants. Imagine that we have a certain deletion of or amplification here, then we are going to have a similar co-expression. It's natural to think that the expression and the co-expression is related to the copy number variation. This is an example copy number variation. So we decided to test this idea using an important region in the chromosome 8, that is the AQ24-3 region. And this, we took 193 copy number variants belonging to the same number of genes, of protein coding genes. And this is how they look. We can have 0 copy number variants until 20 copy number variants. So what we do is essentially to repeat the procedure that was done in the previous work, but conditioning every time with the copy number variation variants of these genes that I already presented. So we have 193 networks, and we had to decide if these networks were different in some way. And one way to do this is to build that distributions that I mentioned, and to measure the differences we see using the Kolmogorov-Smirnov test that is essentially in this way. And we find this. The maximum value is about 0.05, so we can say that there is no difference in this case for the networks, considering the copy number variations of an important region. So we reproduce these networks that are very similar to that I showed previously. They are very similar. It remains the loss of co-expression in cancerous tissues, but now considering the copy number variation. So we have this. This is another way to see the chromosome 8. We start here, we finish here, and in this region we have a lot of contribution or a lot of connections. It's very connected, this region. What this doesn't happen in the control or in the healthy tissues. So this is our main conclusion, is that we take in account the copy number variations of this region, that is important region in breast cancer, AQ24.3, and we didn't find significant contributions in the loss of co-expression patterns. This is the bibliography that we use. Thank you for your attention.
Thanks again, Candelario, for your talk. Now we are going to move for the second lightning talk by Daniela.
Well, first of all, thank you so much for having me. My name is Elena Reyes Gopar, and I am a student in Enrique Hernandez Lemus's lab at INMEGEN in Mexico City. And during the last year, we have been working with Dr. Nixon and Dr. Bendel and their lab in a really cool project where we've been developing a tool called StellarScope to quantify the expression of transposable elements at locus resolution in single cells. So I will start by saying that the reason we're all here is that single cell sequencing has been revolutionizing the study of complex biological systems. So we can now profile tens of thousands of genes simultaneously in tens of thousands of individual cells. And this is all very good, has been providing really interesting information. However, most single cell studies, or all single cell studies, are concentrating on a small percentage of the genome. So the coding DNA sequences, which really amount to about from 1 to 3%, depending on the reference you use, of the whole genome. And the remaining of the genome, the over 90% remaining of the genome, is largely comprised of transposable elements and low complexity repetitive elements. Now, one of the main interests of our project is to study human endogenous retroviruses, which are sequences that live in our genomes. We all have the same set of herbs. These herbs contribute to the complexity of transcriptional regulation, either by donating regulatory sequences or by joining these regulatory networks like the ones Candelario was telling us about. And the fact is that these sequences are actually transcribed and also repressed in some balance that is currently not very well understood, because the expression of these herb sequences has been associated with some disease phenotypes, but the presence and transcription of these sequences has also been associated with perfectly physiological processes. So you can see there are very interesting regions to study, and I hope I can interest you in the tool we've been developing that can be used to study them. So just to be on the same page, herbs are actually the consequence of ancient infections by retroviruses. So a long time ago, some retrovirus infected individuals from a population, and the regular or most known way this works is that these viruses go from one host to other hosts horizontally, so by infecting somatic cells. However, it can also be the case that retroviruses manage to infect a germline cell, and then vertical transmission can occur. So these viral sequences integrate the genome, and throughout evolutionary times and for different reasons, they become fixated on the genome. They aren't purged out. So I think key points about these herbs is that the whole set of herb sequences in the human genome do not actively retrotranspose. All human genomes contain the same set of herb sequences. They are expressed just like any other gene. And I think the true key point is that it's notoriously difficult to quantify these transcripts, even though the material, the nucleic acids, is present in the experiments and also in the data. So I think the most relevant challenge to quantify this the expression of Herb loci is that their sequences are highly similar to each other, and also that they are present in many copies throughout the genome. So, for example, if we have sequencing reads from an RNA experiment, we could have a read like the blue read, for example, that is a multi-mapping read. So it aligns with the same quality to different places on the genome, so to different Herb loci. Or we could, for example, have a case like the purple read that also has multiple alignments. However, there is enough information to discriminate which one is a better alignment and say confidently or more or less confidently that it comes from, in this case, Herb locus number 2 and not locus number 3 or locus number 1. And there is also the easiest case or the kindest case where the yellow read actually aligns to just one of these locus. So it's in the process of the alignment, there is enough information, for example, like a genetic variant, to confidently say that it comes from one place and only one place. And the thing is that most of the sequencing reads that came from Herb loci are actually multi-mapping. And most of these multi-mapping reads, they align equally well to multiple loci. So you can see how this is a problem to be able to quantify them, because how can we know where it actually came from? So what we do is resolve this uncertainty about the position of the ambiguous reads and reassign them to their most probable origin using a Bayesian model. And the way this goes is that we need an alignment as input, so a BAM file to put it in the easiest possible terms. And we also need an annotation of the Herb loci, or an annotation of the transposable elements of interest. This will change depending on what one is studying. In our case, we use Herb annotation because those are the type of TEs that we're interested on. But this stellaroscope can be provided with any annotation that comprises the regions of the genome one is studying. And again, starting from our ambiguous alignment, here we have 3 elements and a variety of sequencing reads. We have the golden reads that provide information because they align uniquely to the genome. You can see that this happens because of variations represented as darker lines on the loci. We also have the dark blue reads, which indicate a probable best alignment. And we also have most of them are actually the light blue reads, which are ambiguous reads. One cannot know where they came from. They align equally well to the 3 sites. And the first step is to calculate an expected assignment weight for each read, which carries the assumption that all of the elements, in this case the three Herbs, are expressed equally. So there isn't any preference in terms of quantity for any of them. Then we use these weights to calculate maximum likelihood estimates, or MLEs, for the proportion of each transcript. So how many reads belong to each transcript. And we go from this estimation. So we change our assumption. We say that we now have a better idea than what we started with about which loci are expressed. So before we thought they were expressed equally, and now we are basing our assumptions on the data. So the true assumption is actually that the quantity of reads, sequencing reads in a library is proportional to the abundance of transcripts in the cell. And what we do is repeat this process multiple times until the algorithm converges. So what this is called is an expectation maximization algorithm, because the process of calculating the likelihood estimates and then updating the weights is repeated many times until we arrive at a most probable quantity. So we end up with final assignments for each fragment. And in a way, this is resolving the ambiguity for each read by borrowing information from neighboring reads, which is different from a typical alignment in that in a typical alignment, each read is treated completely independently. However, using this method, we are able to arrive to resolve these ambiguities and end up with a counts matrix at the level of individual of each individual locus for these elements. And here comes a second challenge associated specifically with single cell data, which is that the amount of reads present in single cell libraries is actually smaller than the amount of reads in the bulk RNA-seq libraries. So our proposal to solve this is that one could go and use a Stellaroscope in what we call a cell-by-cell mode or cell-by-cell reassignment. So for example, in this case, we have two cells, the red cell and the green cell. And these are the reads, the sequencing reads that come from our herb elements from each of the two cells. And we have three T loci or herb loci. And there is a genetic variation which allows to discriminate alignments just for this region, but it's only present in the red cell. So if we do a cell-by-cell reassignment, we would end up resolving the alignments only for the red cell sequencing reads. And that would leave us without any information about the second cell, the green cell. However, we have implemented a Stellaroscope mode called pseudobulk, in which information between the cells can be shared to help discriminate even in the cells where there is not enough information. However, we also have a third mode, which we call bicluster or by cell type assignment, which is based on the assumption that cells belonging to a cell type cluster will share more similarities in terms of their transcriptional profiles as compared to cells from different clusters. So if we were to analyze the ambiguous reads and try to resolve their positions in a pseudobulk Stellaroscope assignment, and that means pulling the sequencing reads from all of the cells together, we might end up with ambiguous reads reassigned to places where there wasn't actually a transcript being produced. So our way to solve that is to do the Stellaroscope reassignments within the cell clusters, so within the cell type clusters. However, this depends highly on the cell type information that users have, which can be of higher or lower quality depending on how one cluster cells and the prior information one has about the cell types. But this is the way, these are the three modes we're providing with the Stellaroscope. And as I think as it's very common in science, there is no one universal solution to solve every situation. So one really has to have information about the system one is studying, the cells one is studying, and the questions one is trying to answer. But just as a demonstration of what
So,
we've been doing.
Elena, we are running out of
Okay.
time, so please conclude. Thank you.
Okay, yes. We analyzed human peripheral blood mononuclear cells with the Stellaroscope, and we quantified their retrotranscriptomes. And I just wanted to show you how relevant it is to be able to resolve these alignments at a locus-specific resolution, because there aren't many tools dealing with these regions of the genome, but the couple tools that exist do it at a family level. So for example, here I am showing you the UMAPs corresponding to 3 HERV-H loci, which are different loci, different parts of the genome. But if you were to quantify at a family level, you would think, you would take them as the same element. And in these UMAPs, we can see that the different loci are actually expressed in different cell types. And this really renders the family level resolution, well, yeah, not so useful when we are dealing with single cell data. So with that, I just hope I can have you interested on this type of data. And yeah, I just want to thank everyone who's been working on this.
So thank you, Elena. Unfortunately, there's no time for questions, but to the attendees, please feel free to add questions then to the chat, and Elena will answer over there. So thank you, Elena. So now it's time for Daniela.
So hello, everyone. Thank you for being here and for this opportunity to share my work. My name is Daniela Senra. I am a physics PhD student at CONICET. And today I'm going to show you some research we've been doing with my advisors, Luis Diambra and Nara Guisoni. We created a software which was implemented in R as an R package. The software aims to quantify the pluripotency from single-cell RNA sequencing data. Typically, identifying stem cells is usually done by stem-based markers. And they are not always available, they depend on the tissue. So we created this method that already includes information from protein-protein interaction networks and gene ontology. This is useful. The application is to use the stem cells identified by our method as a starting point in the trajectory inference. So first, we built a protein-protein interaction network. For that, we used 2 databases, Pathway Commons. We downloaded around 2 million interactions. And on the other hand, we used gene ontology, the genes associated with the cell differentiation process. So we merged these 2 databases, these datasets, and we only kept the interactions, the protein-protein interactions that were part of the cell differentiation process. So when we did that, we obtained this image, this protein-protein interaction network, which is associated with the cell differentiation process that is shown here. So once we have built this network, if we picture just a cell, we have a graph or a network where genes are the nodes, and these nodes are connected by edges. And these edges, we computed them as the multiplication of the expression levels of those genes based on the low mass principle. So if 2 genes are highly expressed, they have higher probability of interactions. So then we sum up all these edges, and this is the score we defined, which was called activity. Here is the adjacency matrix, which has 1s if there is interactions and 0s otherwise, and X is the expression level. So now we have our program. Let's sum up everything. We input an expression matrix, which has to be previously filtered, pre-processed, and normalized. And we input that into our algorithm. And the output is a vector that returns a score, which is the activity for every cell we have. So the higher the activity, the most pluripotent the cell will be. There are other methods to quantify pluripotency. There's a group of methods that are based on statistical and mechanical principles, especially on entropy. The hypothesis of these methods is that undifferentiated cells have high entropy levels, and that allows them to differentiate into many diverse cell types. We use a program that is called an implementation that is called Lancet, and then we used another program which is called C2Trace. In this work, authors found that the gene counts were correlated with the state of differentiation. In general, that's not always the case. So the algorithm finds the genes that correlate with the gene counts and creates a dataset specific gene count signature. This method has a limitation, and it is this limitation is that they are not this is not the best method to identify quiescent stem cells. So let's go to the data set we used. First, we used a human healthy breast tissue data set. We were interested in the epithelial part, which are these ducts here. The epithelial part of the breast is mainly composed of luminal cells and basal cells, and stem cells are located basally. This is important because most of breast tumors arise from these epithelial cells lining the ducts. So we used a data set that is publicly available. It was not ours. And well, we filter, we perform the standard steps, then we normalize, we perform dimensionality reduction, clustering, differential expression, and then annotated the cells. And here are the results. We obtained 3 main clusters. Then we performed differential expression and obtained the gene markers for each clusters. And based on them, we annotated them as basal cells, luminal 1 or luminal precursor cells, and luminal 2 or luminal mature cells. These are related with the hormone response part of the breast. Then we compared all the methods in this data set, the entropy-based one, C2Trace, our method, and an approximation to our method that is quick. This approximation is the same, but the expression matrix used is reduced to the highly variable features. And the results were that our method and the entropy-based one identify the basal cells as the most pluripotent, but C2Trace did not agree with this, as they found as luminal were the highest pluripotent cells, which is not what we expected. Then we tested these 4 methods with other data sets, colon, hematopoietics, and lungs, and we identified that all the methods were positively correlated, being the highest correlation between our method and the entropy-based one. Then we assessed efficiency. Lancet, which is the entropy-based one, was the method that demanded most time. C2Trace was the least demanding time method, and our method was in the middle. Now it's more efficient. Then we also wanted to assess other features like RAM usage. The entropy-based one and C2Trace needed around 6 gigabytes of memory extra, while our method needed around 27 kilobytes of memory. Our method is simple in terms of biological interpretation, and it has the advantage that it can handle zero containing expression matrices, which is important because most of these matrices have lots of zeros, and the entropy-based one cannot handle this type of matrix. So you have to use other normalization techniques. Also, our method is user-friendly. You only need to type 4 lines in R and you can compute activity. So this is an application which I've previously mentioned. Here you can see that the basal cells have the highest activity scores. So we set these basal cells as the root or starting point in the trajectory inference. And we identify 2 lineages going from the basal cells to luminal 1 cells, and from the basal cells to luminal 2 cells. Both lineages go through an intermediate state in the luminal 1 cells. And then after identifying these lineages, we identify the genes that that drive differentiation. So here you can see that some genes are for both lineages, of course. So you can see that some genes are highly expressed at the starting part of the lineage, and other genes at the end part. And the same for lineage 2. Now we're trying this method. We are improving it, and we are trying it with other data sets. Well, if you want to know more about our work, here is the publication. All the data used is publicly available in the GEO database, and the code is also publicly available as an R package in my GitHub profile. So thank you, and if there are any questions, I will gladly answer them.
We are moving for our last lightning talk. Okay, by Isabella Galvão.
So hi everyone, I'm Isabella. I'm a PhD student at the University of Campinas in Brazil. So first and foremost, I would like to thank the symposium for the opportunity to present this work. And what I would like to talk about with you today is the cellular deconvolution of focal cortical dysplasias using single nucleus RNA-seq signatures. So focal cortical dysplasias are a kind of malformation of cortical development characterized by disrupted cortical lamination. It is the #1 cause of intractable epilepsy in children. So children are treated with medication to which they do often not respond. And they require surgical resection of the affected brain tissue in order to obtain seizure control. So it is estimated that up to 50% of children undergoing epilepsy surgery have been diagnosed with some form of FCD. So FCD has several subtypes, but the ones we will focus on here are FCD type 2, which can be further classified into FCD2A and FCD2B, both of which are characterized by abnormal cortical lamination, as well as some abnormal cell types. So both FCD2A and 2B have dysmorphic neurons, and only FCD2B has balloon cells. So the main idea behind a deconvolution algorithm is to estimate cellular composition. So for a deconvolution algorithm, you feed into it an expression matrix derived from a bulk RNA-seq experiment, together with a signature matrix, in our case derived from single-cell experiments. So the signature matrix has the characteristic gene expression of distinct cell types. And the program will try to use this, and through correlation analysis, we'll try to infer what is the cellular composition of that bulk RNA-seq sample. So there are 2 main advantages to this. The first being that there is no need for physical cell isolation because all the analyses are done in silico. This has also been called a digital cytometry. And this kind of approach can be highly accurate, as demonstrated by the high correlations between cellular composition estimated by deconvolution and those inferred from single-cell experiments or immunochemistry. So for this work, we had 2 data sets. The first dataset consisted of 19 bulk RNA-seq samples, 10 of which were FCD2A and 9 of which were FCD2B. So the samples were processed using star for alignment and RSM for quantification to obtain a gene-level expression matrix. For the second dataset, we had an already processed expression matrix of 32 FCD patients, 11 FCD2A and 21 FCD2B. And these expression matrices were fed into the CyberSort X tool to perform the convolution, together with signature matrices or reference files derived from single-cell RNA sequencing experiments. So with this, we tried to estimate the proportions of some major brain cell types in those FCD lesions. So the first thing we did was we performed a clustering analysis, and we saw that independently of the cohorts, so this first line here, which is the city, samples that had the same diagnosis, so either FCD2A or 2B, clustered together. This shows that indeed FCD2A and FCD2B have distinct transcriptomic profiles. Next, we performed the convolution using 4 previously validated signature matrices derived from single nucleus RNA-seq experiments from the adult human brain cortex. So by plotting the goodness of fit of the correlations performed by CyberSort, we saw that CA, the blue here, both for the discovery dataset and for the validations dataset, was the signature that performed best. So moving forward, we chose to focus on this signature for time-related and didactic purposes, but the results obtained with the signature CA were also could also be observed using other signatures. So we first performed the convolution to try to estimate the proportion of major brain cell types, so neurons, both excitatory and inhibitory neurons, astrocytes, oligodendrocytes, microglia, endophilia, and oligodendrocyte precursor cells. So we saw that there's a significant decrease in the neuron proportions in FCD2B compared to FCD2A, this in the discovery dataset. So this decrease is specific for excitatory neurons and not so much for inhibitory neurons. For astrocytes, we also saw an increase in the proportion in FCD2B compared to 2A. These results were corroborated in the validation dataset. So again, we see a decrease of specifically excitatory neurons in FCD2B compared to FCD2A, and an increase in astrocytes in FCD2B compared to FCD2A. These results were also significant when correcting for age or sex covariates. So the next thing we did was we wanted to perform a more detailed subpopulation analysis. So for this, we employed a signature matrix derived from a multiple sclerosis single nucleus RNA sequencing dataset. So this dataset had 22 neuronal and non-neuronal cell types, including some layer-specific cell types, and contained a total of about 50,000 nuclei. So when looking at neuronal populations, results that were observed both in the discovery as well as in the validation datasets was a decrease in excitatory neurons in FCD2B, specifically of excitatory pyramidal neurons. So this happened in both datasets. There was also some other results, such as a decrease in excitatory neurons proportions in FCD2B of neurons in the layers 2 and 3, but this could not be corroborated by the validation dataset. When looking at non-neuronal populations, we again saw a significant increase in astrocyte proportions in FCD2B compared to FCD2A. This is in line with previous analysis done on this work. There were also some other results, such as an apparent increase in microglia in FCD2B compared to FCD2A. And there were also some results which were significant only in one data set. So for instance, the proportion of T cells appears increased in the FCD2B lesions compared to FCD2A, but these results were only marginally significant when we look at the validation data set. And nonetheless, the increase in proportion of the cells is actually quite small. So then we wanted to take a look at specifically some reactive astrocyte markers to assess astrocyte activation. And we can see that for almost all of the reactive astrocyte markers, which are associated with an increased astrocyte function, they are upregulated in FCD2B when compared to 2A. And these reactive astrocyte markers also included some classic ones, such as GFAP, nestin, and vimentin, which are significantly more expressed in FCD2B lesions when compared to FCD2A, both in the discovery and in the validation dataset. So in conclusion, we found that FCD subtypes have distinct cellular compositions as well. FCD2B lesions are characterized by a loss of excitatory neurons, as well as by an astrocyte activation and expansion, indicating a reactive astrogliosis and suggesting the potential role of neuroinflammation in this subtype of the disease. And as for next steps, we are looking at doing an experimental validation, employing GFAP staining, and performing cell counting. So finally, I would like to thank our sponsors for this research, and I would like to thank you for your attention, and please feel free to ask me or email me any questions. Thank you very much.
So thanks again, Isabella. Thank you, Candelario, Daniela, and Elena for your great and inspiring talks. It shows that we have a really promissory future in the field of single cell biology, not just future. This is happening nowadays, and congrats for these great talks by these young researchers. Thank you.
I'd like to comment also on the talks. They were amazing. Congratulations, all 4 of you. Really, really inspiring to see that we already have such nice results. We're gonna see some more tomorrow. And the way we planned for the afternoon is that we have a specific breakout session room for you guys, for the lightning talk speakers, the students and postdocs. So you could discuss more what you have been doing and maybe help out those who are starting and take advantage of more experienced ones. And we will have another breakout session for PIs and researchers who are currently getting funded or who are inspired by the technology and would like to collaborate. So we can discuss specific details about what has been going on since the last meeting and our future plans. So those who like to attend the breakout session meeting now are supposed to click on the links that appear on the chat. So there is one called Trainee Breakout Room. We expect you 4 lightning talk speakers to join there so you can talk. Remember, it's important that it's supposed to be informal, okay? So you can even speak in your original languages when possible and help each other out, like understanding some specific aspects. And another breakout session room for PI and others, researchers and other people who are interested in joining. So there are 2 different links with specific passcodes. So join, and depending on the amount of people who are joining, we will have subgroups within these groups. Okay, we are exactly on time. We plan to join these rooms at 3:25, so you can grab a coffee and then join us for these conversations.